This tutorial on process validation for biopharmaceuticals looking at our drug substance is the common sequence of process validation activities. The information is based on guidance from PDA Technical Report number 42, Process Validation of Protein Manufacturing by the Parental Drug Association, and published in the year 2005. You can visit the PDA's website and purchase that guide at www.pda.org. And our tutorial is based on figure 3.3.1-1 from that guide. To begin this tutorial, let's visually represent the life cycle phases for a protein therapeutic, the drug substance. Looking at the clinical phases, we have clinical trials phase 1, phase 2, phase 3, and then we're into commercial manufacturing, so we have full licensure. I would like to look at the activities that support the common sequence of process validation activities. Let's begin. Let's focus on the supporting activities before, during, and after clinical trials phase 1. So, from development through clinical phase 1, we initiate the process and assay development. We initiate preliminary protein characterization. We establish the preliminary reference standard. We initiate clinical manufacturing, initiate stability studies, and then we're in a position to submit investigational new drug application. Let's look at the parallel process validation activities before, during, and after clinical phase 1. We will perform preliminary clearance of process contaminants. It's very important for our protein therapeutic to initiate viral clearance activities, and following clinical phase 1, prior to clinical phase 2, we will conduct additional virus clearance activities. Now, we're moving into clinical phase 2. Looking at supporting activities, we have now submitted our investigational new drug application. So during the clinical phase period, we will qualify our validator assays. We will establish the protein characterization, establish a process definition, whereupon we can then move into conducting our pivotal clinical trials, which will be phase 3. Now let's look at the process validation activities before, during, and after clinical phase 2. Following phase 1, we will conduct additional viral clearance activities. We will initiate the process characterization studies. We will approve the process validation plan. We will develop and approve the process validation protocols, and this will allow us to move into clinical phase 3, which are our pivotal clinical trials. Now, we enter clinical phase 3. Here, we will conduct our pivotal clinical trials. If successful, we are then in a position to manufacture our conformance lots, submit our registry license application, pass a pre-approval inspection by the registry authorities, and hopefully we will be able to obtain registry approval for commercial manufacturing. We are currently looking at clinical phase 3. If our pivotal clinical trials went well, we're now in a position to execute full-scale validation studies on our commercial manufacturing equipment. We will also execute small-scale validation studies. This would include, for example, contaminant clearance, such as viral clearance, hold time studies for our media and our buffer. We'll then develop our process validation summaries for filing, and hopefully if we do obtain regulatory approval, we're in a position to complete, approve and archive our process validation reports. Now, hopefully, if we obtain regulatory approval, we are into conducting commercial manufacturing and distribution. Throughout this period, we will implement ongoing process monitoring and continuously improve our process. We are now at the licensure phase. Hopefully we've obtained regulatory approval and we're into commercial manufacturing. We have up to this point completed, approved and archived our process validation reports. During commercial manufacturing, we will execute continuous process validation studies and we will forever evaluate the operational parameter range in the spirit of continuous improvements.